That should be better. Sorry, my microphone was super peaky there. I had to reset it. If you haven't done so already, please let me know in the comments that you are here for this. Is it Thursday already? Oh my goodness, time flying. Type something in there. Okay, so at the end of yesterday's class, oh, you guys can't see this. Why isn't this coming up? My iPad and I have a strained relationship recently. Where do we submit our homework doc? Okay, so you don't submit it yet, Caitlin. If you look at the genetic processes unit here on the left, the very, very end of the unit, there's an entry called Midterm Homework Questions Portfolio. That's where you will hand in both of your homework docs from the first unit and from the second unit. So you don't hand it in every unit. You hand it in twice per quad. Once at the end of the second unit, and that's so I can evaluate it for the midterm report, and then once at the end of the quad, and that's so I can evaluate it for your final report card. Do we have any textbook homework from today? Let me look. I'm not 100% sure. Let me take a look. I think most of it is in the course notes for today. I believe it was. We worked on the project in the first block, and then I think everything else was in the notes. Yep. Yeah, everything was in the notes package from yesterday, Kim, so nothing had to go in your homework doc from yesterday. Today, hopefully you guys had an opportunity to watch these two introductory videos. Sorry, these two down here. These two introductory videos into transcription and translation, or protein synthesis. It's the same process. So basically what today is going to be all about is, or at least block one, is going to be reviewing that process in detail. So we're going to go through transcription and translation, and then I'm going to give you some things to work on to practice translating DNA into proteins. You're going to build them, because we know what the rules are for building a protein, and so if we know what the rules are and you know what the rules are, then you can tell me how the protein gets built, depending on the DNA sequence. I mentioned way back in the first unit when we talked about making mRNA vaccines, we can use our knowledge of reading DNA and RNA, mRNA, to figure out what shapes, what proteins a particular bacteria will build, or virus. And we can use that to develop a vaccine, because we know the rules. We know how a cell will take a piece of DNA or RNA and convert it into a protein. So we're going to go through those rules right now. That's what we're going to start with. Then we're going to do a little review of mitosis. So this is a grade 10 review thing as well. Hopefully you covered mitosis. You should have. It's one of the key concepts of grade 10 biology. And then there's a little bit of work for you to do there to do some review on your own. We're going to try and get through all of that during learning block 1 today. We'll see how it goes. We'll do our best. It's a tightly packed day, as I realize. And then in learning block 2, we're going to start our discussion of meiosis. Sounds similar to mitosis, but instead of making an identical copy of a cell, which is what you do in mitosis, you make half cells. I mean, they're full shape. They're still spherical, but they only contain half of the genetic information that a regular cell would have. And you need to do that in order to make a gamete. A gamete is like a sperm or an egg for reproduction purposes. We need to cut our DNA in half and decide which half goes where. So we're going to discuss that process. It's similar to mitosis, which is why we're reviewing mitosis first, but it adds a whole bunch of extra complicated stuff into it. So it's really important to have a good foundational understanding of how mitosis works before that. So if you remember interphase, prophase, metaphase, anaphase, telophase, cytokinesis, IPMATSI, or What were some of the acronyms from last year? In prison. What was it? I have to look it up. There were some really good acronyms last year for mitosis. But anyway, if you remember that process, that would be a real helpful starting point here. But we will review it together. Okay, so that's in general what's on the docket for today. So I'm going to start by talking about protein synthesis. We're going to pick up where we left off in the notes right here. So one little piece of terminology that I'm going to give you to get started with here is that whenever you see the prefix inter, I-N-T-E-R, inter, that means between. So for example, if you have intercellular fluid, that's the fluid in between your cells. You also have intracellular fluid. That's the cytoplasm. That's the fluid inside the cell. So inter means between things, and intra means within things. And you'll see this outside of biology too. This is actually a fairly common prefix. But because they look so similar, inter and intra, people mix these up all the time. Like really, really commonly people mix these up. So this is going to be one of those things that you're going to want to try and set straight in your mind. The difference between inter and intra. So I'll use a chemistry example of this. Intermolecular forces. If you've done chemistry already, grade 11 chem. An intermolecular force is the force between molecules, holding molecules together or repelling molecules from each other. But an intramolecular force is the force that is holding the molecule together. It is the force within the molecule. So those are two completely different concepts, but they sound similar. So just as a heads up, these prefixes are used in biology all the time when we're talking about things that are happening within a cell or between cells or within a process or between processes. So just as a heads up, that's gonna. I always get questions about this. That's gonna be something that you're gonna want to put in your memory. Okay, let's talk again about DNA. Before we get into the actual process of what we're going to do here, I just want to remind you of what the DNA structure actually looks like. So forget my annotations and stuff on here, but just look at this double-stranded piece of DNA. You can't see this. I just realized that this is not on the screen. Okay, hold on a second. That's frustrating. Here we go. So anyway, sorry, I was just talking about this. This wasn't on the screen when I was talking about it either, but inter is between and intra is within. Sorry about that, Cedar. Okay, we've got a double-stranded piece of DNA. Remember that there is this sugar phosphate backbone running across the back. Okay, not really particularly important for this purpose. And then you've got your code that runs along the DNA, and you can just read it. Okay, so I can go across and read the code. T, A, C, G, C, C, A, T. And that's thymine, adenine, cytosine, guanine, cytosine, cytosine, adenine, thymine. Okay, those are the nitrogenous bases, right? That's what you actually read. And what's shown here are two different strands here. We have what's called the coding strand. So the coding strand is shown here on the bottom. I believe that in all of my examples, I draw the coding strand on the bottom. Yes. So the template strand is across the top. Okay. So these two strands have a different purpose. By the way, there is no convention for drawing one on the top or one on the bottom. So usually if this is ever drawn somewhere, it's indicated which one is coding and which one is template strand. Or it will indicate the direction of the strand by saying 5', 3'. I mentioned this very briefly yesterday, this idea of they have ends that are different. And so you know which direction to read it in. But you don't need to know that biochemistry component for grade 11, so I'm not even going to put it up here. I'm just going to suggest to you that I will tell you which one is the template strand and which one is the coding strand, okay? That's for our purposes. In grade 12, you're expected to figure that out, and it's discussed how to figure that out in grade 12. But in grade 11, you don't need to know. I'm just going to tell you. Okay, so here, when I say the coding strand, and I mentioned the coding strand in this case is on the bottom. Okay, so I'm going to label it right there, coding strand. That's my strand on the bottom. I've circled it in green. Okay, that is the actual code. So when you read your DNA, in this case from left to right, that's the way it's arranged here. When you read from left to right, that is the code. If you were to look up, the human genome, which you can do, you can get in a text file, oddly enough, you can read it. That what you're reading is the coding strand. Okay, that is the actual code. The other side, which is called the template strand, is complementary to the coding strand. Okay, we talked a little bit about this yesterday, this idea of complementary, but it has the opposite base pair on it. For error correction, and there's one more really important reason to have a template strand, and we're going to talk about that in a second. But that doesn't, so if you were to read these ones across the top, TAC, GCC, AT, GC, GAA, whatever, that's not the code. That's not what you would record as the code. That's the template. So the template is attached to the code. And in the way I have it drawn here, the code is on the bottom. Okay, have I made that clear? I feel like I made that clear at this point. So the code is on the bottom and the template is on top. So the reason that that's important is because you need to make a copy of the code. Remember that the code is on the bottom. Okay. This code is what you're going to, like I said, make a copy of so that you can build a protein from it. The reason that you need to make a copy and you can't just directly use the DNA itself is that the proteins are made in the cytoplasm, but the DNA is kept in the nucleus. Okay, they're separated, and that's for good reason. In the cytoplasm, there's all kinds of stuff in the cytoplasm. Reactive oxygen species, which are, you may have talked about this in chemistry, I don't know, but they are molecules that have a tendency to react with their surrounding molecules. Like ozone is an example of a reactive oxygen species. And they can potentially damage DNA. Or there may be other enzymes or other things floating around in the cytoplasm that can break down DNA. And those are there intentionally to break down viral DNA. But anyway, there's lots of different things that are harmful to DNA that are in the regular cytoplasm. So under normal circumstances, we keep the DNA separate within the nucleus. Okay, so it's in a separate area and there's no mixing. So in order to read the DNA outside of the nucleus where you make your proteins in the cytoplasm, you need to make a copy of a piece of the DNA and you need to carry that copy out into the cytoplasm. So that process of making a copy is called transcription. And you may have heard the word transcribe before, to transcribe something. Transcribing something just means making a copy of it. So I'm listening to a documentary on ancient civilizations right now. It's like an audiobook. It's really good, actually. And I'm at a part in it right now where they're talking about the library of Alexandria, okay, ancient library in Egypt, where it was like the largest repository of human knowledge in the ancient world. And all of the books that were in it were transcribed from books that passed through Alexandria. So anytime you went to Alexandria, you took your ship there or whatever, they would seize all your books and they would transcribe them all and add them to the library, which is kind of cool. So it was like the biggest library in the world at the time, in the ancient world. And that's transcription. So it's just simply making a copy. Kim, I'm just going to very briefly mention this. The ends of the backbone, the sugar phosphate backbone, have a bonded hydroxyl group, an OH group, in one of two locations, either in the third carbon or the fifth carbon. Okay? And one end of the DNA has it bound on the 5th carbon, and the other end of the DNA has it bound on the 3rd carbon. So we call the 5th carbon one the 5' end, and the 3 carbon one the 3' end. But you do not need to know that for grade 11. So that's what it is. But in terms of like understanding how that influences how DNA is read and the directionality of DNA, you do not need to know that for grade 11. All you need to know is that it's read in one direction. And you can't read it backwards. Obviously, it's gobbledygook if you read it the other way around. Okay? Really, that is all you need to know. You don't need to know anything about 5' or 3'. I'll never mention it. It'll never come up again in the course. So, you just read it in one direction. And I will tell you what direction it is. Like, we're going to read it. I'm going to always show it to you left to right. Okay? And I'm always going to draw the coding strand on the bottom and the template strand on the top every time. So there's no need for you to know that in grade 11. I'm just going to tell you. Anyway, you got to transcribe it first. Okay, so first you make a copy of the code, which is the coding strand at the bottom. You want it to be identical to the coding strand. Then, once you've made that copy, and here is a copy, and I'm going to explain how the copy gets made in a second, then you need to take that RNA, that's RNA by the way, the copy is made out of RNA, a slightly different molecule than DNA. It's very similar, but it's slightly different. But it is only half, obviously. It's not a double-sided molecule like DNA that has 2 backbones. There's only 1 backbone in RNA, which is actually perfect because it makes it so it can fit through the tiny holes in the nucleus and get out of the nucleus. So the nucleus has pores in it. They're called nuclear pores. And single-stranded DNA, like RNA, can fit through the pores. But double-stranded DNA cannot fit through the pores. So the DNA is kind of trapped in the nucleus, whereas these can penetrate through the nucleus and go out into the cytoplasm. Okay, so they're single-stranded. And then once you've made that copy of the code, the last part then is to read the code and actually build the protein, build the thing out of the code. And that second process is called translation. And that's because it's translating a signal. It's taking a RNA signal and making a protein. So it's translating it from one medium to another. That's why it's called translation. And so sometimes these processes together are called transcription and translation. And sometimes it's just called protein synthesis because that is because that's what you're doing. You're synthesizing or building proteins. So the last part is getting translated, and then it gets turned into a polypeptide, a sequence of amino acids. Okay, and we'll get to that in a sec, how that works. If you watched the video yesterday, you've already seen this process at work. I'm just talking about some of the details here in a little bit more detail. So, excuse me. Going back, how do we actually make a copy? Well, it's all because of this complementarity between the base pairs. So I mentioned that if you look across from the coding strand, the base pair that's matched up with it is always the complementary base pair. So T's always pair with A's and C's always pair with G's. So what actually happens is you unzip your DNA. Okay, an enzyme comes by and breaks all those bonds in the center of the ladder. So it just zips by. In grade 12, you actually talk about what those enzymes are called and stuff. You don't need to know that for grade 11. Okay, so an enzyme comes zipping by, splits it in half, and another enzyme will come by and attach to a start codon. Okay, so the start codon, if you look on the coding side, which would be down here, this is the coding side, right, is ATG. Anytime you see an ATG in DNA, that's the beginning of a protein. Okay, it's the start of a new protein. So that's called a start codon. And the enzymes are pre-programmed to always go attach to the start, which is exactly what they do. So they attach to that start codon. If you're looking on the complementary side, on the template, the code that you're looking for is TAC, of course, because it is the complement to ATG. Okay, so the enzyme will come, and it only attaches to the template. The actual code itself is completely ignored. Okay, which seems strange, but it'll make sense in a second. Because what it does is, as it runs, as that protein, I'm going to draw it on here, as the protein runs along, the enzyme runs along the template strand, what it does is it grabs always the complementary base pair for each of these letters as it goes across, which is really the whole reason that you have that complementary template strand. Because at the end, if you do that, so I'm gonna go through and do that right now. So it runs across, and because of the chemical bonds, and this is purely a chemical process, it adds in the complementary one to all of these. C pairs with G, C pairs with G, A pairs with T, T pairs with with one exception. Important exception. RNA, just to make things a little bit more complicated, RNA does not have thymine in it. So RNA is what we're building on here. And specifically, we're building mRNA. mRNA stands for messenger RNA because it's the message that goes from the nucleus to the ribosome. Okay, that's mRNA. So that's what we're going to be building. I think I wrote it off to the side here. The message itself is called mRNA. Okay, so we got to build the message. But there are no thymines in ribonucleic acid, in RNA. So instead of building thymines, everywhere you would normally see a T, you're actually going to see what's called a U, a uracil. Okay? Why this extra level of complication? I don't know. But this is the way things have evolved in living things. There's a slightly different nitrogenous base that pairs up with A. It is uracil. Okay? So it's going to go A, U, G, C, G, G, and then this would be another U here because it would be a T. A, G goes with C, A goes with T, which becomes uracil, A goes with T, which becomes uracil, T goes with A. Okay? So that enzyme zipped across, and it built a complementary strand to the template. Okay? And what you'll notice is that if you build a complementary strand to the template, that is exactly the same as the code, which is what you wanted, right? You wanted to build a copy of the code, with the only difference being that T's are now U's. Okay, and that's because of RNA. There are no T's in RNA, so it had to switch them to U's. But other than that, the actual message that you built is exactly the same as the coding strand. Perfect. So we've made a copy of the message of the code, and now that code is going to leave, okay, and go out into the cytoplasm, and then here's the actual code. So this is just a copy of it down here. So there's the code. Yes, Anastasia, yes I can, because all the enzyme is doing is taking a base pair that is complementary to the template and helping it to attach to the template and make that complementary sequence. Those base pairs are just floating around inside the nucleus, and eventually they would bind with their complementary base pair. But the process would take forever. Because of this enzyme, it can zip across there and build one of these things in like milliseconds. It's so fast. It just zips across and builds this sequence off of it. Actually, if you go on YouTube, there are real-time simulations of this where you can actually see what it looks like in real time. Maybe I can find one of those videos in a bit, or I can post one. but it's an extremely fast process. And so they attach and they just zip across and find all the chemicals, the complementary base pairs that would chemically bind, and just zip, attach them all real quick. Now, when I say that an enzyme is a catalyst, it is. It speeds up the rate of a chemical reaction, but it also makes chemical reactions happen that would otherwise never happen, or in an order that would never happen naturally. So, for example, enzymes can be used to do chemical reactions that require energy. So in the cell, you're not going to get a reaction happening between two things that requires energy to occur, because there's no, I mean, I guess they could potentially happen just by chance. They had the right collision energy or whatever when they bumped into each other to cause a reaction. But in realistic terms, it would never happen. That chemical reaction would never happen. For example, a lot of times when enzymes are binding two things together, putting things together, that chemical binding would never happen under normal circumstances in the cell. It's too energetically unlikely for that energy to exist at the right place at the right time for those two things to bind. But what enzymes can do is they can use ATP, which is the chemical, the energy chemical of cells. It's made from sugar. The mitochondria makes ATP, by the way. And it can use up ATP and force chemical reactions to occur that otherwise would never happen, that are basically that go against the normal flow of energy. So normally, well, you guys already know this. Exothermic reactions, ones that give off energy, are far more common than endothermic ones, because they're favorable for them to occur energetically. And so in order to force the reaction to occur, enzymes allow you to put energy in and basically make it go in a direction that it normally wouldn't go. So I don't want to give you the wrong impression about how enzymes work. They do operate like catalysts, but they also allow chemical reactions to take place that never would take place, just because of how they function. Okay, so it's kind of like a type of catalyst, a subtype of catalyst. Okay, so now we've got this message. Okay, AUG, CGG, UAC, UUA. Okay, that is mRNA. We've made this messenger, and it still has a start codon at the beginning. That is the codon that says beginning of protein, build here. But now in mRNA, the start codon looks a little bit different because it's been translated into mRNA. Now it's AUG instead of ATG. So, I mean, it's the same codon, but it's what the codon looks like in RNA. It looks like an AUG. Okay, so we've got this messenger, and then that messenger is going to leave the nucleus and go into the cytoplasm, where it's going to encounter the protein builders. And hopefully from your review of cell parts yesterday, you will know that the protein builders are ribosomes. Ribosomes are very unusual organelles, because they are also made of RNA. Confusing. So ribosomes are made out of two pieces of RNA that are sandwiched together. And when I say RNA, I mean, yes, exactly the same as RNA like this, but just a specific sequence of RNA. And they are sandwiched together to make like it looks like a sandwich. It looks like a little hamburger, a tiny little hamburger. And that type of RNA that makes up ribosomes is called rRNA. So the messenger, that is what you're sending from the nucleus to the ribosome, is called mRNA, messenger RNA. And the ribosome itself is made of rRNA, ribosomal RNA. And I think at this point, you might be noticing that, well, we use RNA for a lot of things. There's actually one other type of RNA that's involved in this process called tRNA, and we'll get to that in a second. But wow, RNA has like a bunch of different roles. It can store a message. It can translate a message. It can move one chemical from one place to another and bind it to a specific other thing, which we're going to talk about in a second. That's cool. RNA does a lot of things. That is why RNA is thought to be the original biological molecule, or at least one of the reasons why it's thought to be the original biological molecule, because unlike DNA, RNA can have a whole bunch of different jobs that are all related to making copies of things. And so there's a good chance that the original organisms were RNA-based instead of DNA-based, like all living things are now. And the reasoning for that is that it could be both the message and the translator of the message all in one type of molecule. So it's far more likely that a simpler organism had a completely RNA-based system instead of a DNA-based system. Something to think about. I think that's cool. Where was I going with this? We've done the translation. Right. No, we haven't done the translation. We're doing the translation. So we've got our RNA, our mRNA. We're going to attach it to a ribosome, okay? And then the ribosome is going to run along the RNA, and it's actually going to translate it. It's going to convert it into amino acids. So amino acids are the building blocks for proteins. And you would have seen that in the videos you watched yesterday. But proteins are made out of a sequence of amino acids. Amino acids are the smallest chunk of a protein, the Lego bricks of proteins. And there are a bunch of them. Here they are on a table down here. I'll show you how to read the table in a second. But these are the amino acids that all proteins are built out of. Okay, so they're just sequences of these proteins. Oh, there's a ribosome on the right there. So the purple that I've highlighted at the bottom, that's the ribosomal RNA. It doesn't actually show the rRNA because that would be confusing, because it'd be RNA on RNA on RNA, and then it starts to get confusing. But right through the center of this, the orange part is this middle part right here, the orange, is the mRNA. So that's the message that's coming from the nucleus. So in comes that message from the nucleus. It comes down, gets locked into one of these ribosomes, and then it's going to run through the ribosome. And every time it gets to a 3-letter sequence, okay, so it reads it 3 letters at a time. Those 3 letters, by the way, are called a codon. I mentioned the start codon here. The start codon is the beginning codon, the first code in the sequence. Okay, by the way, lots of new vocabulary here. Codon. add that. So the start codon is the first 3-letter sequence that begins the translation. Okay, and then after that, you're going to read it 3 letters at a time. So you're going to read start, and then the next 3, and then the next 3, and then the next 3. So it reads it 3 letters at a time. So, for example, the start codon is AUG. The ribosome reads the AUG, and I have the table for reading it down here. When you read the table, you start in the middle. Okay? So we're going to start in the middle. And it was AUG, right? Yeah, AUG. So you start in the middle, A. Then U is the next one. You work your way outward from the middle. A, U, and then G. Okay? And then if you go to the outside of the ring, it will tell you which amino acid that codes for. So that is what the ribosome is going to add to the protein. So AUG is methionine. And methionine is short formed with MET. Okay? Now, I also called that the start codon. I wrote at the top here that that one is the start. That means that every single protein starts with the same amino acid. So the same chemical is found at the very beginning of every protein, and it is. So every protein begins with a methionine at the very start. Now, sometimes that methionine gets trimmed off by the Golgi apparatus or by the ER, actually, during the folding, or there's like some processing that happens after the protein is built. But at least it starts there. So there is a methionine at the beginning every time. So there it is. Methionine gets added in. And you'll notice that there's a little thing attached to the methionine down there called a tRNA. tRNA, also made of RNA, is called transfer RNA because it is what takes the methionine and adds it to the codon. Okay? So every amino acid that's floating around in your cytoplasm is attached to a tRNA. And the tRNAs are what allow you to link up the right amino acid with the right codon. So the tRNA, going over to my diagram at the bottom, has a little code on it. Okay? And that code is complementary to a specific code on the RNA, the mRNA. And so that's what allows it to stick to the right thing on the RNA, and then on the other end up here, it will bind the amino acid that's attached in sequence to the protein that's getting built. Okay? Another thing that you might notice about this process, because it's like letters matching to letters, and it's all chemistry. This is just a chemical process happening. It's things that chemically bind to other things, binding to them. That's it. And when you really get down to it on the cellular level, when you really dig in deep and look at how things work, it's all chemistry. Every single thing that happens in there is a chemical reaction, which is really fascinating. I mean, that's something that I think when you start your career in biology, when you really start at the very beginning, you don't really understand that everything is chemistry. But when you dig into these processes, everything is chemical on the cell level. And that's biochemistry. So if you're really interested in that chemistry and how it works, the grade 12 biology course is for you. You do a ton of biochemistry in that course. And that's when you start getting into the real meat and potatoes of like how all of this actually works and how we can control it in the lab. But it is just a chemical process. So there's no magic happening here, which I think is fascinating. Okay. So we've gone through and we've read the first sequence, AUG, and then the ribosome has added methionine. And by the way, those tRNAs just break off. Once the amino acids get added, the tRNAs just fall off, and they go and attach to another of the same amino acid so they can get used over and over again. They get cycled. So tRNA goes away. And then now it's going to read the next three letters in our mRNA. So the next three letters are CGG. Okay, then we go back down to our chart and go, all right, CGG. CGG. Well, CGG is arginine, short form ARG. Okay, so it's going to add an arginine. Then it's going to read the next three letters, the next codon, which is UAC. You go down to your chart. UAC. That's a tyrosine, short form TYR. And there it is. You add it in. And then you look at the next three letters, the next codon, UUA. You read it off here. UUA is leucine, and then it adds in a leucine. Okay? And as this is going, it's adding more and more amino acids. They are sticking to each other. The amino acids have bonding sites on them as well. They're different shapes, and they stick to each other, and they make their own little shapes as they come out. And that's part of how the finished functional protein gets formed, is that it's like the product of all of the different parts of all the different amino acids sticking to each other in a certain way and making a specific shape. So they showed this in the video yesterday, but this idea that you can basically build any shape that you want, and they're not just shapes, they're like shapes that have sites for chemical reactions and things like that on them. But you can build whatever shape you want just by putting in the correct sequence of amino acids, and that's how you can get all these different shapes just through your DNA code. So just in very brief here, then to reiterate, you start with your DNA, which is double-sided. You unzip it, and you make a complementary strand to the template. Okay, in this case, the template is shown on top, and you go through and you make that complementary RNA, mRNA, to the DNA. So it'd be A, U, G, etc. Okay, then that mRNA strand is going to go out of the nucleus and attach to a ribosome. And the ribosome is going to go through and read it 3 letters at a time, and it's going to add an amino acid for each 3-letter sequence, which is called a codon. Okay, methionine, arginine, tyrosine, leucine. And it's going to go on and on and on and on and on until it gets to a specific sequence called a stop codon. And I should mention, by the way, that these stop codons and start codons and stuff are the ones that are on here are specific to humans. These are human start and stop codons. They can potentially be different for different species. I'm pretty sure they're common for all eukaryotes, but I think prokaryotes have a different system. So just anyway, something to point out really briefly. But a stop codon, for example, would be like UAG or UAA or UGA. Okay, those are known as stop codons. And the stop codon is the opposite of a start codon. Whenever it gets to a stop codon, it stops, and then the protein is done. Okay, so there's no stop codon in the DNA sequence until it stops, until it's the end of the sequence. Okay, if you have a stop codon at the end of a protein chain, do you still include it? There is no actual, there is no amino acid associated with the stop codon. It doesn't have an amino acid associated with it. So there is nothing to include when you hit the stop codon. So the last amino acid in the chain would be the one before the stop codon. So whatever is the last one before the stop codon is the actual end. And then the stop codon just means let go. Like it actually tells the ribosome to let go of the protein. It's finished. I hope that answered your question, Dominic. Lillian says, so when you unzip the DNA, does the DNA become RNA? The DNA does not become RNA. It's chemically the same when it's unzipped. And once you release that enzyme that builds the RNA, another enzyme comes by and rezips it. It puts it back together again. And that's to protect the DNA. So the DNA is protected while it's zipped up. There aren't any of these exposed bonding sites just like sitting out and about. You don't want to like leave those exposed. So another enzyme comes by and zips it back together again, like just like a zipper. Sorry, it's rezipped. Yep, it's absolutely rezipped to protect it. Okay, let me see if I actually covered here everything that's said here. Double check. In the nucleus, DNA acts as a template to produce messenger RNA. Okay, we talked about that. Not all your DNA is read at once. No. So you only read certain sequences at once. I mentioned this yesterday, that certain cells are going to be reading certain parts of the DNA to make the appropriate proteins for that cell. For example, let's say you're a melanocyte in your skin. Well, you have to read the sequence to make melanin, which is the colored component that makes your skin darker. Everyone has some melanin in their skin unless they have albinism. And so that sequence is being read and converted into melanin, the protein melanin. Whereas if you are a corneal cell, I don't know why I keep using cornea as an example, but it's a very different cell. Like in the surface of your eye, that sequence that makes melanin is never being read. There is no melanin in the cornea of your eye, and so that it's basically turned off, that particular sequence. So not everything's being read at once. Some things are being read far more than others, depending on the type of cell. And that's controlled by the introns, the non-coding part of the DNA. We already mentioned this idea that sequence that makes one specific protein is called a gene. So we're talking about reading genes here. When producing mRNA, the DNA is read and a slightly different instruction set is made for the ribosome. Yeah, that's the mRNA. And I already mentioned this, the idea that all thymines are replaced with uracil in RNA. That's just a chemistry thing. mRNA leaves the nucleus, moves to the rough ER. Oh, so this is important to note. I actually bypassed this. Where this is occurring in in the cytoplasm, okay, where the actual translation is occurring, and that's where the mRNA is being converted into a protein. Some of it is happening just in the cytoplasm. So you would remember this also from your review from last year, which is that some of these ribosomes are just floating around in the cytoplasm. The ribosomes that are floating in the cytoplasm are producing intracellular proteins. Okay, so going back to the beginning here where I talked about inter and intra, that's why I mentioned it. Intracellular means proteins that are going to stay in the cell, within cell. Okay, so if it's just a free-floating ribosome that is making the protein, that protein is going to stay inside the cell. However, a lot of the proteins that cells make are actually meant to go outside of the cell. They're meant to act somewhere else or be a chemical messenger or whatever. Like insulin is a great example. Pancreas makes insulin. Pancreatic beta cells make insulin. And that insulin needs to be shipped out of the cell. So in order to ship it out, you have to build the protein with a ribosome that is attached to the endoplasmic reticulum. So that's called the rough ER. You may have remembered that. The rough ER has ribosomes stuck to it. And the reason for that is because after you make it, so I'm just going to quickly draw it. Let's say you've got a ribosome, and then you've got your rough ER, okay, which is like a tube. What happens is that when you build that protein, okay, it's going to have to be packaged up with a bunch of other proteins, usually by the Golgi. And so in order to corral all of those proteins together that you've just built, you actually build the proteins into the ER. And the ER is going to transport that protein through the ER. And it actually does some folding and some other things too. You don't need to know for grade 11. And it's going to transport it to the Golgi, where the Golgi is going to put this protein into a vesicle. So a vesicle is just basically like a vacuole. It's just like a sac that is inside the cell. So it's going to put the protein into a sac, and it's going to put all of the same proteins that are being built also into that vesicle. And that way, when you need to release that vesicle into the extracellular fluid, or otherwise known as the intercellular fluid, okay, these are intercellular proteins that we're talking about now, you can release them all at once by just pushing that vesicle out to the surface of the cell and then merging it with the cell's surface, which will push those proteins out of the cell. So there's two ways that this can go. You can either be built by a free-floating ribosome, which I should probably write this down actually. A free-floating ribosomes produce, I'm going to write it. Remember that intracellular means within the cell. And then rough ER ribosomes produce intercellular proteins. And those are proteins that are meant to go outside of the cell. This comes up a little bit here because I'm mostly going to be talking about the intercellular protein route, which is the bottom one here, where the rough ER is involved. And the reason I focus on that one is because it has a bunch of extra steps. So the other one, the free-floating ribosome one, is the same process all the way up until once the protein is translated. Once the protein is translated in an intracellular protein, the ones that are meant to be used in the cell, it's done. The protein detaches, it's in the cell, it's doing its job. But if it's an intercellular protein, there are some more steps that are added to the process to package it and prepare it to release it from the cell. And so I'm going to talk about that process. But if you're thinking, well, what is the other process like? It just ends when the protein is done, once it's done being assembled by the ribosome. Okay. that was a lot. So I should mention that this process right here of how we take some DNA and make it into a thing, make it into an actual functional Lego, a functional biological unit like a protein where it does work inside of the cell or outside of the cell, is the most important concept in all of biology. Okay, so what you've just learned is known as the central dogma of biology. It is the most important concept in all of biology. There is no area of biology where this is not important. Okay? This is important literally every part of biology. DNA transcription and translation is an important thing for you to know and understand how it works for everything. Evolution, biochemistry, molecular biology, even ecology. Really, any area of biology is reliant on an understanding of this particular fact. All of modern biology is built on this. So anytime you hear about some discovery happening, new medicine, it's almost always something that's related to a deeper understanding or use of the central dogma of biology, which is transcription and translation. So, and I really mean this. If there's only one process that you take out of this course, you go and you're like, I forget about taxonomy. I don't know about that. I forgot about meiosis. I forgot about how to predict which genes are going to get given to a particular offspring. I forgot about how evolution works. Whatever. Don't forget this. This will come up in every biology course you ever take. So anyway, just as a heads up, this is the central dog. It's the most important concept in biology, which is there is an element of complexity to this. I agree, but it really is crucial that you have at least a basic understanding of how this process works. It's really important. So that being said, I'm going to go through it one more time, okay, but just in more basic terms, okay? Keep in mind that I am discussing here for an intercellular protein. So these are for proteins that are going to leave the cell. And that process is just a little bit longer, okay? It adds a couple extra steps at the end. So this process, by the way, below here, skips over the first part. It skips over the transcription part. So before you even get to this summary down here, DNA is going to be read by an enzyme. Name of the enzyme not important for grade 11. And it's going to make a messenger RNA, an mRNA, on the template strand. Okay? And that will be virtually identical to the coding strand of DNA. So that would be the code. All right? That code is going to get sent, the RNA code is going to get sent to the rough ER. Okay, so that's mRNA. It gets sent to the rough ER, to a ribosome. It's then going to get fed through the ribosome, and it's going to make what's called a polypeptide. Originally, and this particular note I borrowed from another teacher, but what it says there, I think in your note as well, is it says protein. It's not actually a protein at that point yet. So it's called a protein once that full sequence of amino acids is done, and then it gets folded into its final shape. Okay, so sometimes that requires some modification by the Golgi or the ER. Those things do some additional folding. Often, sometimes they even snip out pieces of the polypeptide to make the final protein. Once it's done all of those final modifications, then it is called a protein. Okay, so it's not quite a protein at this point. All that the word polypeptide means is amino acids linked together. Those amino acids are also called peptides, And a poly just means many. You might probably know that from math, like a polygon. So polypeptide just means many amino acids. So a polypeptide is a bunch of amino acids linked together. They're not a protein yet until they're finished being folded. Okay, and sometimes trimmed, but usually just folding. Before it's a protein, it's called a polypeptide. That is the name of it. Right there, polypeptide. So at this point in the sequence, it hasn't done any of that final modification. Yes, yeah, exactly. So we would still call it a polypeptide. Okay, so then the newly created polypeptide then gets moved through the rough ER. Okay, so it's actually going to get transported through the ER. That's one of the functions of the ER, is to transport molecules around the cell. And it will pinch off into what's called a vesicle. Vesicles are essentially little balloons. They're made of lipids, exactly the same material that the cell membrane is made out of. So they're just like tiny little cell membranes, little bubbles of cell membrane that contain packets of proteins inside them. Okay, so those are vesicles. And then the vesicle gets transported to the Golgi apparatus. And when you were doing your review, or maybe you remember from last year, that the Golgi apparatus is responsible for modification and packing of proteins. That's its main job in the cell. It has other functions as well, but its main function is to pack proteins, package them up for export, to send them out of the cell. So those proteins are going to go to the Golgi, and then the Golgi is going to do the final folding and trimming and adjustment to the polypeptide. So here it says the protein is folded. Again, this should say the polypeptide is folded. I understand the confusion here. I'm going to try and be as specific as possible, because it's really not called a protein at this point. Make that smaller. Okay, so the polypeptide gets folded into a 3D complex shape specific to the function of that particular protein, whatever it is, and then it gets repackaged into another vesicle. So it gets put back into another package of completed proteins. Now it's a protein. Now we call that a protein. So it gets pinched off from the Golgi, and it gets sent to the cell membrane. Now, I should mention here that it's not just getting sent to the cell membrane all the time for no reason. There's actually a trigger, usually a chemical messenger, that tells it to send the vesicle to the cell membrane. So, for example, I mentioned that your pancreatic cells make insulin. Insulin is made in this exact process, okay? And then it's put into a vesicle by the Golgi. But you don't actually release the insulin from those cells until you eat sugar. When a person eats sugar, that causes a binding of that sugar to the outside of those beta cells in the pancreas, and that activates a series of chemical reactions which will cause this vesicle to be sent to the surface of the cell and released into the blood. And then that's what increases the insulin in your blood, which will then bind to a whole bunch of different cells in your body, telling them to absorb the sugar out of your blood. But you don't actually release the insulin until it's time. It's not like you're constantly just dumping the insulin into your blood. You only release it when it's appropriate to release it. And that really goes for all of these proteins. They're only released when they're needed. Okay, and then after that, the protein goes and does its job, whatever that is. It could be an enzyme, it could be digestive enzyme, it could be a messenger protein, it could be anything. There's a million uses for proteins. So they go into circulation. So that's the basics. Sounds like it's not the basics. Sounds like it's a lot of detail. But that is actually the basics. What you talk about in grade 12 is the names of all of the different chemical messengers and enzymes that do these things in this process, and you need to know those for grade 12. So that's when it really gets complicated. This is a broad overview of the process. But again, those things are good to know because we can control them. We can control this process and make it work for us, which is why this is such an amazing process that we've figured out here. Like, can you imagine somebody in the lab figured this out? They actually figured out which 3-letter combinations use which amino acids. I mean, that's groundbreaking. That's amazing. And because of that, we can look at a sequence of DNA and we can decide what it makes. I can't stress how important that is. Every advancement in biology, cellular, micro, everything that is reliant on this understanding. I mean, this is like the great leap forward in understanding biology. I can't emphasize it enough. Anyway, I mentioned here at the bottom that it's important to note that the cell needs a specific protein to help with a specific cellular process or function within the cell. Then the ribosomes that are floating freely in the cytoplasm produce it. Okay? In other words, it's not related to the ER. So the ER are not involved in the production of intracellular proteins. And those proteins just begin their work when they enter the cytoplasm. So that's just for the other types of ones at the bottom. Okay, what am I going to get you to do here with this? I'm going to get you to practice the process of transcription and translation and using this, okay, which is also known as a protein building chart. I'm going to get you to practice using, I think it's pretty incredible too, Ryan. It's amazing. I'm going to get you to practice using this protein building chart to figure out how to build specific amino acid sequences or polypeptides from specific sequences of DNA. Okay? So what I'm going to give you here is the template strand. Now, it says fill in the DNA top strand. Again, this is not my worksheet, but there's no such thing as the top strand. That's not. There's no tops and bottoms in DNA. So what it actually means is fill in the coding strand. The coding strand. What you're always being given in each of these examples is the template strand. So you're given the template. The template, remember, is what you build your RNA off of. So it's ignoring the coding strand in each of these examples. But I'm going to get you to add in the coding strand. And just to keep it consistent with our example above, because where was it? With our example above, you'll notice that we did the coding strand here on the bottom and we did the template strand on the top. I'm going to keep that consistent. Every example you ever see from me, I will keep this consistent so that it's not confusing. In previous iterations of this course, I've seen all kinds of stuff, worksheets where they keep flipping which one is template and which one is coding, and it is like insanely confusing for students. So I'm going to keep it always the same. If you see it on the top, it's going to be the template. If you see it on the bottom, it's going to be the coding. Okay, I'm going to keep that consistent. There is no actual method for which way to write it. As I mentioned, there is a way to figure out which is which by the chemical bonding on the ends of the DNA, but you can't see that in any of these examples, so there's no way for you to determine it. So I'm going to always keep it the same. So where are we here? So I've given you the template strand in each of these cases, and I'm going to get you to go through and start by writing in the coding strand underneath. Okay, so I've done it in purple here, and remember that the coding strand is complementary to the template strand. So ATG, C binds with G, G binds with C, C binds with G, T binds with A, C binds with G, C binds with G, G binds with C. That understanding, by the way, that A always binds with T and C always binds with G, you must know this. I know I mentioned that yesterday, but lock that information away. It is pivotal that you know that those base pairs always bind with each other. So lock that information away. I'm going to get you to go through and write the coding strand first. Then I'm going to get you to decide, well, what would the mRNA sequence look like from this particular strand of DNA? Remember that you build the mRNA from the template. So the top here is the template, and you're going to build it from the template, which means that it's going to be complementary to the template. So when I say the template, remember that I'm talking about the top strand here, okay? That's the template. So then you're going to go through and build the complementary strand to the template, but anytime that you would say T, you're going to switch it with U. That's the only thing you got to remember when you're doing mRNA, is that all of the T's become U's. So here we've got C binds with G, G binds with C, C binds with G, T binds with A, C binds with G, C binds with G, G binds with C, C binds with G, C binds with G, G binds with C, T binds with A, C binds with G, G binds with C, A binds with, and this is where you would say T, except it becomes U. Okay? So that's the only thing that you got to watch out for, is those circumstances where it would be a T, but now it's going to become a U because it's mRNA. Okay, so that's the transcription part. That's making a copy of the code. All right? The last part then is to convert that code into a polypeptide, into a sequence of amino acids. So you read it 3 at a time. The first one here, AUG, is also known as the start codon. I think I had the presence of mind to make the first 3 of all of these a start codon. It should be. And the last sequence should always be a stop codon. I hope I did that. I'm not 100% sure whether I did that, but the last one should be a stop codon. Anyway, AUG is methionine. It's always the first one. You'll notice it's always the first one. GCG. Okay, let's go up to the chart. If you want to look up the protein building chart, if you just want to Google it, I mean, this is obviously a common thing in biology. You can just Google this chart. There's various forms of it. I like the circle one best, but if you want to have it in another window while you're doing this decoding, it's easier than jumping back and forth through this PDF. But it's totally up to you how you want to do it. But see, like, I don't even remember what the sequence was that I just read. It was GCG. Okay, so we're going to go GCG. So GCG is alanine, short form to ALA. Okay, ALA. Okay, so that's one amino acid. And now we look at the next 3, which is the next codon, AGG. AGG is arginine, short form ARG. Okay, and etc. Keep reading it along until you have the entire amino acid sequence. Keep in mind that the stop codon does not code for an amino acid, and so the previous amino acid is the last one in the sequence, and the last one is just stop. You can write stop if you want, but it's not an actual amino acid. One quick thing to note here, and I did already mention this. Sometimes I forget to mention this, so I put this in here as a reminder to myself. The DNA cannot leave the nucleus. It's double-stranded and doesn't fit through the nuclear pores. It's too big. That is the reasoning, or at least one of the reasons, why you have to have an mRNA sequence to carry the message from the nucleus out into the cytoplasm. That, and it's dangerous in the cytoplasm for the DNA. So, anyway, the amino acids, where do they come from? Oh, I also mentioned this here, and we're going to talk about this in more detail in the animal systems unit. Where do you get all those amino acids? You eat them. This is the reason that you have to consume protein in your diet, because you need your own amino acids to build your proteins for your body. So you have to eat protein. and break it down into amino acids as part of your digestive process. So that's where they come from. They're floating around in your cytoplasm of all your cells, but you have to eat them. So you have to eat protein. Some of these, I give you the DNA, and you tell me the mRNA and the amino acids. Some of them, I give you the mRNA, and you tell me the DNA and the amino acids. And some of them, I give you the amino acids, and you give me the other two. Okay? So keep in mind that if you're going backwards, let's say that you're starting with an amino acid sequence and you're going backwards to the DNA. There is more than one potential sequence here. There's more than one potential mRNA that makes the same amino acid. So you get to pick one. You can pick whichever sequence of mRNA you want to fit there. It really doesn't matter. They're all the same. They all do the same thing. And so your DNA will be different. It won't be exactly the same as mine depending on the mRNA that you choose. And that's for the ones where you're going backwards from the protein, from the polypeptide, back to the DNA. Okay? So if you look below, there are some examples of that. I'm going to get you to try this entire worksheet. I will post this. I believe it's, well, I'll post it again, but it's in the course notes folder. So if you want to check your work when you're done, I would highly recommend it. Double check to make sure that it's accurate. With one caveat, don't forget that if you've gone backwards from amino acid to these two, and I just mentioned this, that you may have a different sequence from mine, but as long as it works, it's fine. So it may not be exactly the same as mine. What happens if you have too much protein in your diet? What happens is your cell metabolism attempts to convert the additional protein into energy molecules. So you don't need the amino acids for building. They are extraneous. They're surplus. So you actually convert them into sugars, is what you do, or fats. So they go through a really interesting biochemical side chain where they are converted into energy molecules to use as a supplemental form of energy. In that process, you have to remove a bunch of the extra nitrogen from them, which you urinate out. That becomes additional ammonia in your urine, uric acid and ammonia. But the bottom line is they become energy. So if you have a whole bunch of extra protein in your diet and you are also getting enough energy in your diet by consuming the appropriate amount of carbohydrates and fat, you will convert the additional protein into fat and store it. If you're using the energy, like you're using up all the energy from the energy that you're consuming, you will just burn that additional energy as well. So it just becomes part of your regular energy metabolism. If you consume way, way too much protein, a very high amount, a lot of using that side chain can be unhealthy in the long term. In the short term, it probably doesn't have too many negative effects. But you go into a body state called ketosis. You may have heard of this before. It's the same thing that like the ketogenic diet is based on. If you've heard of people like doing keto before, and that is when protein is being used primarily as an energy source. You're using it as to build proteins, but you're also using it a lot for energy because you've removed some of the fat and carbohydrate intake from your diet. It's mostly carbohydrate, actually. You've mostly removed the carbohydrate intake, and so you're using protein to supplement that. If you consume too little protein, then in order for you to get enough amino acids to build all the things that your cells need, you have to start disassembling proteins that are already part of your cells. The biggest store of proteins in your cells is in your muscles. So what happens if you don't get enough protein is you start breaking down your muscle fibers in order to get enough proteins to just build the structures of life, just to keep your cells functioning properly. And so you get what's called muscle wasting. Muscle wasting is when your muscles go away. They waste away because you use the proteins in them to support the cellular functions of your cells. And eventually, if if you don't have any protein and you lose the majority of the protein in your muscles, it can cause death. It causes disease and death eventually. You do have to have some protein intake in order to survive. So, but you can survive long periods of starvation, or at least lack of protein through breaking down your muscle fibers, which is what our ancestors did. Over the winter, a lot of the time, they did not have sufficient protein intake, and so they pillaged their own muscle proteins, and it's a survival mechanism for humans. Okay, I'm going to let you work on this. Let me quickly look. Oh yeah, sorry. There's actually two components to this. I'm gonna get you to work on this. Okay, and the second part is I'm gonna get you to try and summarize this. Okay, so this is the process right here. You'll see it. I've numbered it. I've numbered the locations that are involved in this process. And what I want you to do is, in your own words, use those numbers and summarize this process from beginning to end in your own words. Okay? Make an attempt at it. And this will very much help you remember the process. If you can, and you can go back and look through your notes as you do it, but if you can summarize it yourself, what's happening at each step, that will be very beneficial in terms of helping you remember this process. Okay? So I'm going to get you to do that as well. The next part, the cell cycle part, I think we're going to do that at the beginning of block 2. So I'm going to let you work on this right up until 10:20. It takes a little bit. It will take a little bit, especially the writing out the process in your own words. It will take a minute. If you have some extra time, there is a really cool cell visualization. I posted a link to it here for protein synthesis. What this is a simulation of what these molecules would look like based on our understanding of how they are built chemically. So really, this is what the process looks like. It's not these colors. The colors are added in for your to make it easier to see stuff. But this is the process of transcription and translation, but just kind of colored up. But this is what it actually looks like. This is a visual of what it actually looks like inside. So if you're curious to see what, so for example, there is a ribosome right there. The green guys right there are ribosomes. The blue parts right there, I believe, are mRNA that are being read by the ribosomes. I may be incorrect. Those could be the completed polypeptides that are coming out. If you look, some of these ribosomes are attached to ER. There's a ribosome attached to the ER. Oh, see, I am wrong. So the blue part right here is the mRNA being read by the ribosome. And the pink part that is poking out into the ER here is the protein that is being built by the ribosome. So it's being built into the ER for transport. This is a nuclear pore, which allows mRNA to transmit between the nucleus and the cytoplasm. And then on the inside here, you can see some DNA being split apart and some enzymes coming along and building RNA molecules off of those DNA molecules. So this is an actual visualization of what it does look like inside of your cells as this process is happening. Anyway, if you're interested, that's there. You can I can't do it on my iPad, but you can click on the things and it tells you what each thing is as part of the process. Some of those things will be things you've never heard of and we don't talk about till grade 12, but some of them are things that we've just mentioned. The last thing I have for you to do, again, if you have extra time, this is not essential for you to do, is there's an article that I found this last quad, about basically what a functional use for this, how we have actually determined the shape using supercomputers actually of a finished protein based on our knowledge of this process of transcription and translation. So and it talks about the use of that, what we actually use that for, knowing the structure of these proteins. So if you're interested, you can read that article. It's relatively recent, and it's written in pretty much lay terms, so just for like the average reader. So there's not a lot of highly technical language. It's a BBC article, so it's not an academic paper or anything like that. But anyway, it's a great it's a great article about a functional use of this understanding. Okay, there were some questions here. I won't forget about them. Do some fruits and veggies have protein? Yes, they do. Plants have proteins as well. Depending on the vegetable, some have more and some have less. Avocados have a moderate amount of protein, but other things like apples have very, very little, almost unmeasurable amounts of protein in them. There are protein, but they're not a sufficient source of protein for you. You require fairly large quantities, grams of protein per day. And so a lot of fruits and vegetables are not great storers of protein. Things like legumes, beans, have a lot more protein per gram of bean. Beans especially are very high. Nuts are often very high in protein depending on the nut. And so yes, there are plant proteins, but not all fruits and vegetables contain high quantities of protein. So it's really specific ones that are high in protein. Most of your protein, unless you're a vegetarian, likely comes from eating meat, which is mostly protein. But obviously there are lots of people that don't eat meat and they get theirs primarily from vegetables. What happens if you don't consume enough carbohydrate? You go into another type of starvation. It's not protein starvation, but essentially you start using other things for your primary energy source, mostly fats. If you're consuming enough fat, that's usually a fairly low consequence because you can use fat as an energy source, no problem. If you are not consuming enough fat or carbohydrate, then you have to start using protein exclusively as an energy source, and that can be problematic depending on how much protein. You don't want to have 100% energy from protein in your diet. That's not healthy. A mix of protein and fat is not terrible. Low carbohydrate is surprisingly fine. I was always raised that you had to have a balance between protein, carbohydrate, and fat, but you really don't require that much carbohydrate. And there's a lot of evidence coming out suggesting that, well, certainly as North Americans, we consume far, far too much carbohydrate. Simple sugars in general. We love our sugars. We consume a lot of sugar. And there are a lot of negative consequences of consuming too much sugar. One of them is we have a epidemic of obesity, and it's mostly from sugar consumption, not fat consumption, which is a huge misunderstanding that people have, that eating fat somehow makes you fat. It's all about total calories, not about how much, not about eating fat making you fat. That's not how that works. Anyway, we're going way off topic. If you have specific dietary questions, like questions about diet stuff, we're going to do a whole day on that when we talk about animal systems. So I'm going to come back to that point if possible, if you have more questions about that. Yes, it does look like an acid trip. I agree. That's just the colors that they chose, but it's very complex inside. Do cell organelles actually have color, or are they just shown like that? They do not have color. So most of these would be the same, either clear or relatively uninteresting browns. If you've looked at cells under the microscope, you will find that they really don't have too much color going on the inside. So all of that, all the color that you normally see is either a stain that they've applied to make things certain colors, to make them easier to view under the microscope, or they've been added later with computers, which is exactly what you're seeing in that image. It's a computerized image where the colors have been added later, but they are not fantastically colorful like that example. It'd be kind of cool if they were, but that would also mean that you would be fantastically colorful. You were made of these things, right? But if you can imagine, like people that have albinism, so that they don't make a lot of melanin, they are very white. Their skin is very white, almost has a transparent-like quality to it, because most of your cell's components don't really have any color to them. Red blood cells do. And that's because of the iron that's contained within their hemoglobin. Again, we're going to address that on a later day. But most cell components don't really have a color. Okay, I hope I answered all your questions. I'm going to let you get to work on that. So we're doing everything all the way up to where it says Part 2 Mitosis Review. Some of that stuff's optional, as I mentioned, but if you have any questions while you're going through, you're like, oh, this doesn't make sense, I don't know how to do this, don't forget to post in the chat. And if you need to, we can jump into a Google Meet and I can quickly show you or go over a specific example with you that'll hopefully clear things up. Okay, I hope that was informative for you. And I'm gonna, like I said, I'm gonna let you work right through. So I will see you at 11:05. Okay. Let's start off. First of all, did anybody have any difficulty with the activity DNA to mRNA to amino acid, or in any direction, the protein synthesis practice, or potentially writing it in your own words? As usual, if anybody says anything in the comments, I'll circle back. What we're going to do now is do a review of mitosis. This should look familiar to you since it was covered in grade 10 biology. Sure. Which one would you like me to go over? I can do just pick one of the questions. Which one did you find the most challenging? That question was for you, Nate, by the way. Which one did you want me to go over? Just let me know. I'll pick one and I'll go through the entire thing. Okay, yeah, sure, absolutely. Just so you know that it is posted. So the course notes are posted, and this is completed, posted. But I can absolutely just go through an example for you. Absolutely, I'll do the first one. So I did part of it before the break here, but I'll keep going. So you have the template strand. You were given the template strand, and then you just go across first underneath and make the coding strand. Okay, so ATG, GCG, AGG, G, and then I'm going to continue there. Complementary to G, or to C is G, C is G, G is C, T is A, C is G, G is C, A is T, C is G, A is T, A is T, T is A, A is T, C is G, C G, A T, C G, T A. Okay, so that's the screen is black. It was just working a second ago. Thanks for letting me know, by the way. Okay, here we go. Okay, so we've got, I started with this example earlier, and I'm just going to continue. Again, I'm writing the complementary ones because I'm writing the coding strand here below the template. Okay, so this is the part that makes up the actual code. So all I'm doing is writing the complementary base pairs here. Oh, sorry, that one's wrong. G is C, T is A, C is G, G is C, A is T, C is G, A is T, A is T, T is A, A is T, C is G, C is G, A is T, C is G, T is A. Okay, those are just the complementary base pairs. Okay, give me 1 second, Callum. I'm gonna finish this example, and then I'll look at the one in number 4. It's possible that I made a mistake in the code that I put there, but let me double check. Okay, so now that I've got my template and the coding strand, I'm gonna build my mRNA, but I'm gonna build it off of the template, which means that it's gonna be complementary to the template, and it's going to be the same as the coding strand. But the only difference is that from the coding strand, all the T's will be switched to U's. Okay, noted, Callum. So there's 2 ways you can do this. You could go through and build it as complementary to the template strand. So just go through and add the complementary base pairs, but all the T's would be switched for U's. Or if you've already made the coding strand, you could just make it the same as the coding strand, because that's also complementary, but just switch all the T's for U's. So that's the same thing. So I'm going to keep going through here, and I'm just going to write the same as the coding strand, but switch all of the T's for U's. But keep in mind that it's built on the template strand. That's how you get this code. Okay, so G, T's or U's, U, G, G, U, G, A. Okay, so I've gone through and it's the same as the coding strand, but all the T's are switched for U's. And that's the same thing as saying it's complementary to the template strand. And that's how it's actually built. It's built on the template strand, which is why the template strand is given above always, because you build the mRNA off of the template. Okay, so now we have our mRNA, and now we have to go through and check each of these codons 3 letters at a time. So we have AUG was a methionine, GCG was alanine, AGG was arginine. Now we have CGG. I'll go up and check CGG. Just erase this stuff on here to make it less confusing. Okay, so we've got CGG. CGG is arginine, ARG. And then we've got CAG is the next codon. I'll switch colors here for each of these. CAG, which is glutamine, which is GLN is the short form. Then CUG. Yeah, CUG, which is leucine, so that's LEU. Then UUA is the next one. UUA, which is leucine. And yes, there are multiple codons that code for the same amino acid, so that's not a mistake. The sound in the background is a baby crying. It's my son. And earlier it was my other son crying, so I'm not sure what's going on up there. There are some sad children. It's my son. The next one is UGG. UGG, which is tryptophan, TRP. And then the last one is UGA, which I'm just going to take a wild guess here is the stop codon. Erase this so you can see it clear here. UGA, stop. Okay, and you can write stop STP, but I mean it's implied it's the end. So okay, some interesting things to note about this. One thing is that there, like I said, there are multiple codons that code for the same thing. So that has a little bit of error protection built into it. You'll notice that multiple of the last codon, the third codon, usually code for the same thing. And what that means is that often the third letter can be mutated, can mutate. That is, it's been disrupted by a chemical or by whatever reactive oxygen species. And so you've mistaken the last letter in a codon. It can be a different letter and still make the same protein at the end. So there is an element of error protection there, which is a pretty interesting evolutionary trait. So that exists. And the other thing to note is that the worst possible thing that you could get is to get a letter inserted into the sequence, a bonus letter. For example, if in our DNA we added in a letter here, okay, that can happen. That's the worst kind of mutation, because what that does is it shifts all of the codons after that over by one letter, and which makes them all incorrect. It makes everything after that spot in the DNA incorrect. That's called a frame shift mutation, because you're shifting the frame that you're reading on by one letter. And it's also called a nonsense mutation because it makes everything after it nonsense. It makes it do nothing. So that breaks the entire rest of the gene permanently in that cell. So that is the worst kind of mutation that you can get, is a frame shift, because it breaks that particular protein. Luckily, the mechanism behind how you get a frame shift mutation is fairly rare. I believe it is the rarest type of mutation, so it's not as problematic, but it does happen. Okay, I'm hoping that explained. How was that, Nate? Did that satisfy you in terms of how to check over the process? It's too bad there's so much delay. I think it's like 3 or 5 seconds. It should be less, but I have it on the lowest latency setting. So, okay, cool. All right, I'm going to keep going. So, like I said, we're going to review the process of mitosis. Remember that the purpose of that process is to build an exact copy of your DNA and separate out that copy, where one copy is going to go to one daughter cell and the other copy goes to the other daughter cell. We've just gone over in terms of how complex DNA is and how long it is. So it's not the simplest process. You have to make an exact copy of your DNA, hopefully with no errors, although you always get a few. And then you need to subdivide that DNA so that 46 pieces go in one direction and 46 pieces go in the other direction, and you need one of each chromosome to go in each direction, obviously, so that both daughter cells are identical to the original. So I realized that when you did this last year, I'm not even sure if you talked about the number of chromosomes that are actually in there. Obviously, they don't show you all of them. When you see like a visualization of this, they only show you 3 or 2 or something. So it's a lot more complicated than it seems on the surface, because we have so much DNA in there, so many pieces. Anyway, you want to pass them on to a daughter cell. Things to keep in mind, and this hopefully is all review for you, that the cell cycle is mostly interphase. So that's what's shown here in pink, and interphase is when the cell is just doing its cell job. The only thing that's happening here that's related to mitosis that we care about is that during the S phase, you're making your copy of the DNA. That's called DNA synthesis. Synthesis means to build, or it's a chemical reaction where you build. So you're building DNA during the synthesis. There are enzymes that do that. They run along the DNA and build a copy of the DNA. Again, using the complementarity of DNA, which is, again, useful that it's complementary. So it goes through and builds an exact copy. And then in G2, the last phase, gap 2, you make several of the structures that you need for cell division. So you're building centrioles. You are building any of the cellular machinery that will be required for mitosis. But other than that, the cell is just doing its job. So the DNA synthesis is actually just happening in the background while your nerve cells are doing nerve stuff, conducting nerve impulses, and your muscle cells are contracting and whatever. Like, this is just happening in the background. And most of your cells are in interphase at any given time. The vast majority of your cells are in interphase. If you ever look at a cell sample from somebody and you see that many of the cells, a large percentage of the cells, are not in interphase, that is one of the indicators that you may be looking at cancerous tissue that is multiplying far too much, because the mechanisms within the cell that restrict cell division are broken. So anyway, then you get in. Then the mitotic phase begins. It's relatively short. It takes about, I think for most eukaryotic cells, it takes about a day. It might be 12 hours. For prokaryotic cells, it's considerably shorter, so it takes about an hour usually to go through the whole process. For prokaryotes, it's much simpler. And then cytokinesis is the actual splitting, which kind of begins during mitosis and then it ends at the end. Okay, so I already mentioned this here, this idea that you need to make, during interphase, you need to make copies of all of your homologous chromosomes. Remember that you have 23 types of chromosomes, and then each one has a pair. Okay, those are called homologs. And you have one from mom and one from dad. Those are the maternal chromosomes and the paternal. So maternal from mom and paternal from dad. Pater being father and mater being mother in Latin. So here they are. Again, here's a full karyotype for a human. And all I've done is just color-coded the first few of them here. You've got a maternal and paternal for each. Okay? And those are homologous chromosomes. Then, during the mitotic process, or I should say, sorry, during S phase, so that, remember, this is during the regular part of the cell's life where it's just doing its cell job. During the S phase of that, of interphase, you make a copy of each of these. And the copy is going to be attached to its copy. The two copies are attached to each other at the centromere. So if we have a look here at what the same karyotype looks like after you've done that, it looks like this. And I should mention, by the way, that this karyotype here is for a male. Notice it has an XY at the end. I couldn't find a complete karyotype that has the copies for a male. This is a female, so that's a minor difference here. But this is the same karyotype other than that difference, except now there are copies of each, and the copies are attached to each other at a centromere. Okay, so we've got the male chromosome 1 now has a copy of it attached, and the maternal chromosome 1 has a copy attached to it. And the same thing for 2. You've got your 2 paternals, and you've got your 2 maternals. Okay, they're attached to a centromere. This is always confusing for people because now these two versions that are beside each other, so the 2 blue ones here, those aren't homologs to each other. The blue and the pink here for chromosome 1 are homologs. They contain the same genes, but the 2 blue ones are identical to each other. They're exact copies. So the name for that in genetics is we call those sisters. Sister chromatids are exact copies. So the number one thing that people confuse during this process in grade 10 and grade 11 is that they mix up homologs and sisters. So homologs are, again, the same genes. They code for the same things, but they're not identical to each other. You have one from your mom and one from your dad, and they're not identical. Sisters are identical to each other. So now we've made identical copies of each of those. And so the blue ones here would be sisters to each other, and the pink ones here would be sisters to each other because they're exact copies of each other. Okay? So it's important to keep those two concepts separate. Homologs and sisters are not the same thing. All right. So, I think I already mentioned what's mentioned under here. During S phase, you make duplicates. So now, if you were to actually go through and count each stick, keeping in mind that there are now identical copies attached, now there are 92 sticks of DNA, or 92 strings of DNA, because we had 46 chromosomes, right? And then we made a copy of each, so there's now 92. We don't say that there are 92 chromosomes, though. This is always confusing for students as well. There's still 46 chromosomes here, and that's because when you say that something is a chromosome, you're saying that it is unique genetic information. 46 unique strips. But because the sisters are identical, you don't say that now there are 92 chromosomes, because the chromosome implies unique genetic information. So there's still 46 chromosomes. There are 92 chromatids. Chromatids are the names for the actual pieces of DNA. Once you wind them up into this visible form, remember that during interphase they're unwound and just like kind of laying out like spaghetti. But during prophase, you wind them up into sticks that you can see. That's what makes them visible under the microscope, and they look like this. And then they're called chromatids. So there are 92 chromatids here, but still only 46 chromosomes. This may be some terminology that has passed you by in grade 10, because this is frequently misunderstood by students. So you just have to be careful with the idea of chromatids and chromosomes. Okay, so we have 46 sister chromatids. There's 92 total chromatids, but there are 46 pairs, okay, that are linked together. 46 pairs of sisters. Okay, and now the sisters are ready to enter prophase at that point. So I'm going to go through the entire phase from beginning to end. If you were in my class in grade 10, you'll notice that this is the exact same note. And that's intentional. I'm trying to keep it as consistent as possible because this is all review. So, again, interphase consists of 3 parts. G1, which is gap 1, which is just growth. The cell getting bigger, making new cell parts. That's it. The middle part is synthesis, the synthesis phase. Sometimes I wish my office had a door. It's awfully loud up there. The synthesis phase is in the middle when you reproduce the DNA, when you make a copy of the DNA. And the last part here is still growth. So the cell is continuing to grow and function as normal. But you do make additional centrioles, which are used for separating out the DNA, which you may remember from the last time you went through mitosis. So centrioles are structures that create these spindle fibers that pull the pieces of DNA. So that's what's being shown over here. Both of these cells are considered to be diploid. Remember that diploid means has the full set of genetic information. So it's also shown as 2N. So 2N means the full set of genetic information for a cell. Once you copy it, you don't call that 4N. You would think, oh, well, that's 2N, and then you made another 2N. That's 4N. Except you didn't increase the amount of genetic information because you've only made identical copies. It would be like if you had a book and then you had the identical book. You wouldn't have 2 times the information. You would just have a copy of the same book. Okay? So I like to call that 2 times 2N or double 2N, but it's not 4N. Because if you write it as 4N, that means double the amount of information. And it isn't. It's an identical copy. Okay? So keeping in mind that the DNA at this point is not in the form of chromatids. It is in the form of chromatin. Chromatin means that the DNA is all unwound and just like laying out in the nucleus. And if you look at this under a microscope, it doesn't look like anything. It just looks like a dark area. Absolutely. Absolutely, I can. Okay, so hopefully that was okay, Michaela. So you've doubled the DNA. You've made identical copies. It's still in its chromatin form, so still unwound. But at that point, now it's ready to actually start mitosis. You've made all the DNA and all the structures that you need to do mitosis, and now you're ready to go into the mitotic process. Okay, so now mitosis actually begins, and the first phase is called prophase. In prophase, you take that DNA that was just laying out, see if I can draw it here, just laying out as a long strip of DNA, and you actually start to wind it around these little proteins called histones. Okay, so you wind it around these little proteins called histones. And that is what actually makes the DNA visible under the microscope. So the unwound form is called chromatin, and the wound form is called a chromatid. So we've wound the chromatin into chromatids. Another way of saying that is we say that they have been condensed. It's condensation. We start to make spindle fibers at that point, which you can see over there. And you probably didn't talk about this in grade 10, but the inside of the cell has structures inside that actually hold the cell shape. It's like a scaffolding inside the cell. It's called a cytoskeleton. It's just a bunch of proteins that go across the cell. And obviously those would get in the way. If you can imagine, if the cell is just like full of scaffolding holding the cell in a shape, if you leave that there, the DNA is going to get caught up on it and stuck to it as you're trying to organize it. So the cytoskeleton goes away. That's not super important for you to know for this course, but I just mention it because it is in fact happening. And the centrioles begin to migrate towards opposite ends of the cell. I mentioned the word centrosome here. You see that in the literature a lot. It's mentioned in the textbook, and people are always like, is it called centriole or centrosome? Two centrioles together are called a centrosome. Okay, and you have sets of centrosomes that go to either side of the cell. So they're used often interchangeably, but two centrioles is called a centrosome. So what you're actually seeing in that diagram over there are centrosomes going to opposite ends. But again, it means the same thing, really. It's like the difference between Golgi body and Golgi apparatus. The different layers of the Golgi apparatus are called Golgi bodies. But when you put all the bodies together, that's an apparatus. You may be wondering why you see both of those terms for cells as well. That's the difference between the two of them. Okay, so we've got the nucleus breaking down. Did I leave nucleus breaking down under there? I didn't even mention it. Cytoskeleton breaks down. Spindle fibers form. Huh, I didn't put nucleus breaks down. Why didn't I? That's weird. Let me add that. So the nucleus dissolves or breaks down as well. Hopefully this is review for you. I'm hoping it's review. I realized that the end of the year was a little bit shady last year, so you may have missed some stuff. If you do miss something here, please be aware that this note is posted. So if you go in the class notes folder, this will be in there. I'll have to update it now because I forgot to add in the nucleus breakdown. I don't know why, but I will update it at the end. But this note is available completed. Okay, so that's prophase. Metaphase is next, and prophase is the most complicated, obviously. The most is going on during prophase. During metaphase, fairly simple here. There is a structure in the center of the cell called the Metaphase Plate, or sometimes called the equatorial plate, depending on what source you cite. And the chromosomes are lined up on the plate. In other words, the plate goes in between the two sisters. So the sisters are actually sitting on opposite sides of the plate. And it's a physical structure. It is there, but you can't see it. It's transparent, and under the microscope, it's not visible. But it is sitting underneath the centromeres. So that's metaphase. Fairly straightforward. Then the centrosomes start to reel in the spindle fibers. So they have these little motor proteins. They reel in the spindle fibers and yank on the centromeres, which break in half. And then the two sisters will go in opposite directions, keeping in mind that you should have 46 pairs of sisters. So there's going to be 46 chromatids going one way and 46 chromatids going the other way. That's what happens in anaphase. And if you've been in my class before, you know that I like to call anaphase banana phase. This helps me remember what phase it is, because the X's turn into bananas. I don't know, it works for me. Helps me remember what anaphase is. And I actually, in my mind, especially if I haven't done this for a while, I work backwards in either direction from anaphase. I know the order of the phases. And if I know banana phase is when the X's turn to bananas, so then the stage before that must be where they line up, and the stage before that must be when the nucleus breaks down and the chromosomes form, or the chromatids form, rather, from chromatin. This is also during anaphase when cytokinesis begins. This is a little bit confusing because we normally talk about cytokinesis happening after mitosis is done at the end, but it actually starts right around anaphase. And you can see in that diagram there where I added it in that a cleavage furrow begins to form at this time. So a cleavage furrow, what's happening there is that the cell builds a belt made out of proteins that go, most stuff is made out of proteins, a belt that goes around the outside of the cell, and then another protein comes along and starts cinching in the belt. So it starts making the belt shorter and shorter and shorter and shorter, and what that does is it eventually will cause the cell to split into two. So it pulls the edges of the cells in to cause them to compress the middle, and then eventually causing them to split into two. So that's anaphase. Anaphase, you got the beginning of the cleavage furrow and the chromatids being pulled apart. And then telophase, which is the last part of mitosis before actual cytokinesis occurs, before the cells actually separate. The chromosomes are going to cluster to the opposite poles. They will decondense and go back into their chromatin form. That's their spaghetti form, where they're just kind of laid out. And the nuclear envelope is going to reform around them, placing them in a safe and secure nucleus. And you'll notice that the cleavage furrow continues to expand during telophase. And then the last component of this is that you continue to pull in on that belt that goes around, the metaphase belt that goes around, and eventually you're actually going to get a split. That's the true cytokinesis, is the splitting of the cell from what was one continuous cell membrane into two. And you end up with two identical cells to the cell that you started with, hopefully, if you did it right. And in a normal plant and animal cell, takes about 10 to 20 hours. It actually depends on the cell type, so there is a range, but it is far, far shorter for prokaryotes. And for protists and fungus, I have no idea. I would assume it's probably similar. It's probably similar. Although I have no experience with that, so I don't know. one thing that I mentioned at the bottom here is that what we've shown here is really only one type of chromosome, or you could think of it as two chromosomes being separated out here. So we've got, actually that's not true, there's four, because we would have, go through and colorize them here. Let's say that this is chromosome 1, okay, from dad, paternal chromosome 1. And we've got chromosome 1 from mom. And then we've got chromosome 2 from dad. And then we've got chromosome 2 from mom. Okay, so this is only showing an organism with a 2n number of 2, a diploid number, a 2n number of 4, rather, sorry, maybe a 1n number of 2, where there are only 4 different chromosomes in this organism. In a human, there would be 46 of these. Okay, it would be 2n is equal to 46. This is only depicting a 2n equals 4 organism, which is, I'm sure it exists. I'm sure there's an organism that only has 4 chromosomes, but there are not many. So that's a pretty small complement for a eukaryotic organism. So something to keep in mind, there would be far, far more of these at the end. And another thing to keep in mind, when we started this, there were 4 chromosomes in the original organism, and each cell at the end is also going to have 4. They will have the identical number. It's the same. So you're making an exact copy of your cell. Okay. How are we feeling about that? Hopefully that's review for you from last year. I'm trying to decide whether I want to start the next part right now or whether I should just give you a chance to do this. Let me take a look. Let me take a look at how much stuff I could actually reasonably get done. We have about an hour, but I think I might want to give you this time to do the work for this part. Yeah. Yeah. I'm going to start meiosis tomorrow. I think that's too much stuff to do in one day. So, and there's a little bit less tomorrow, so I think that'll work out perfectly. What I'm going to do is, oops. I'm going to get you to watch this summary video of mitosis. Okay? I highly recommend that. Is this on the screen? I'm hoping this is actually showing up. Yes. Watch this summary video of mitosis. There are some questions that go along here with the from cell division, asking you about what is happening in each of the phases and a little bit of connecting stuff to the DNA stuff that we have been talking about. Okay, so that's these review questions. There are 5 questions in total. Okay. And then the last part here is a video from Khan Academy, and it talks about why cells have to divide. You may have wondered why cells even do this. Like, what's the point? There are single-celled organisms. They do all the things. There are protists that have, what's essentially a mouth and digestive area and whatever. It's just one cell. So why didn't we evolve to just be really big cells as opposed to multicellular organisms that have a whole bunch of different cells. Like, what's the advantage to making more cells? And it has to do with surface area to volume ratio. And so I'm going to get you to watch this video on surface area to volume ratio, and it talks about the exact answer to that question. And you're going to need that information in order to answer the very last question here from the homework, which is why do cells divide instead of just getting bigger? We're going to talk about this in a little bit tomorrow. We'll do a little recap. But I want you to see if you can try and answer that question on your own after watching that video. Okay, so I'm going to get you to do that. And then the very last thing that I'm going to get you to do today, I know there's always so many things. These days are so busy, is I'm going to get you to just watch this video as an introduction to meiosis. Normally we would actually start meiosis today, but I think it's too much. Watch this video. It's a great summary. And while you're watching it, I want you to think about what the difference is between mitosis, which we just reviewed, designed to make exact copies of cells, and meiosis, which its purpose is to make gametes, to make sperm and egg. How are those two processes similar, and how are those two processes different? I just want you to think about it. You don't need to write anything down, but as you watch this video and it goes through the process, think about how those two processes are similar, because connecting the two is going to make it a lot easier for you to understand meiosis. And this will serve as a good introduction before we really get into detail with the process tomorrow. Okay, so before we get started, does anybody have any questions about what you're going to do? We're not going to do the note on meiosis. That I'm definitely going to do tomorrow, because that's too much stuff for one day. But so the idea here is that you're going to watch this summary of mitosis, complete the review questions in the book. You'll need to watch this Khan Academy video on surface area to volume ratio in order to answer the last review question, unless you already know the answer. So if you remember, I do talk about it in grade 10, so if you remember it from last year, that's totally fine. But if you don't, this is a great video explaining that concept. And then you're just going to watch this as an introduction. Okay, I don't think that's too much work for an hour. You can probably do that in an hour, no problem. So you're becoming more efficient, right? We're doing our best. I know this environment is not ideal. Okay. If you have any questions, post them in the chat. Send me an email. Let me know where you're struggling. If you have any questions about the assignments that are coming due at the end at midnight tonight, you want to discuss a potential alternate deadline because you need an extension, again, let me know, guys. Don't be that person that just disappears into the night. If you don't think you're going to hand it in on time, talk to me and let's work something out, okay? Let's try and be responsible about it. That's part of the expectation for this course. Is the stream stopping? Yes, it is. I mean, I'll be here. It's going to continue as stream, and the chat will be there, but I just so you're not watching a video of me working on my other course, I will put up the other screen. No, there's nothing you add to your homework document from this. It's interesting. This unit is actually fairly light on the homework questions for the homework doc. There are a few, but not a ton. Many of the other units have a lot more textbook stuff. So this one, I stick more to the notes. Part of the reason behind that is I don't feel like the questions in the textbook are great for this unit. So I kind of circumvented the text a little bit and wrote my own stuff. I just feel like that they're a lot more focused and more directed at what the curriculum is asking you to know. So, do we have to add anything so far? Okay, well, let me look. That's easy enough to figure out. Nope. Nope, nothing from yesterday. And I'm almost certain there was nothing today. Nope. Not yet. Not yet. There are some later, though, for sure. Okay. Again, if you need anything, guys, let me know. If you're working on the projects, you want any feedback on anything you've completed so far, give me a heads up.